Okay, so our next presenter is Dr. Ronan Mac Loughlin, currently Associate Director of Science and Emerging Technologies in Aerogen. You've got more than 20 years of experience in designing and developing aerosol delivery systems and drug device combinations, primarily in acute and critical care settings. Dr. Mac Loughlin has authored more than 90 research articles, 180 presentations and conference papers, as well as various patents. Currently working, has a position of Adjunct Associate Professor in Trinity College Dublin's Pharmaceutics and Pharmaceutical Technology departments, as well as a range of other roles as well, and is also co-chair of the Paediatric and CF Working Group of ISAM. Today, Dr. Mac Loughlin is going to talk to us about aerosol deposition in a mechanically ventilated ex vivo porcine lung. Thank you very much. Over to you.
Thank you. I'm not hooked up to be mobile like Wilbur, but I'll have to stand here. So good afternoon, and thank you very much for the introduction. So mechanical ventilation is not something probably most of the world concerned themselves with until COVID, certainly, when we all knew what a mechanical ventilator was, because all of a sudden became unavailable and we were worried about global supply of mechanical ventilators. But they are used on a day-to-day basis in the ICU and critical care settings. And they're used as a life support system, whether for the critically ill patient or often many times just during operations such as surgical operations. So mechanical ventilation and aerosol drug delivery, as I'll describe very quickly, is a thing. It happens in real life, but there's still a huge amount left to understand about the space. So we've covered the conflicts already, but again, mechanical ventilators themselves have been around a very long time. Many would have heard of the iron lungs of old, and this is what the ICUs in the polio pandemic a number of years back looked like. Those iron lungs where it was negative pressure ventilation. They sucked air out to allow your diaphragm raise for you to take a breath in. But things have advanced on, and they were quite rough, right? They weren't delivering a pre-titered breath volume at set pressures with any great accuracy. But now, because these systems are being used with premature infants, for example, kids that'll fit into the palm of your hands, you need to be very, very careful. But of course, it's the full range of patients from small to large. And more generally, the system itself is comprised of the mechanical ventilator itself. The air that passes from that passes over a humidifier down the inspiratory limb into the patient's lung, where then the exhale and the air goes out. Obviously, you're going to use the inspiratory phase to carry your aerosol into the patient's lungs. Now, there's a lot of different devices currently used in mechanical ventilation. Global surveys of clinical practice for aerosol therapy in the ICU would suggest it's everything from PMDI to jet nebuliser and vibrating mesh. And we can see that about 70% of healthcare professionals use aerosol therapy during mechanical ventilation. So again, that's a very big number of patients, and it's a very big number of drugs as well that are nebulized. And the reason I'm telling you this is simply to set the scene that this is something that happens on a regular basis. And often, we'll hear about the DPIs and nebulizers for home care applications and spontaneous breathing patients. But there's a huge population of patients that require mechanical ventilation and concurrent aerosol therapy. So with that, the real considerations are patient safety. Things like jet nebulizers, for example, wouldn't be suited for use with premature infants or just infants because of that driving gas that's required could overinflate the lungs, leading to the risk of barotrauma or volutrauma. But there's obviously now, we're all very familiar with healthcare professional safety as well, the clinician safety, where you don't want what has been known about for years, the sputum shower, the worst named thing you could imagine. Essentially, when they're maintaining the ventilator circuit, patient secretions will have worked their way through the circuit. When you break that pressurized circuit, there is a sputum shower, and that's not something, certainly post-COVID, that anybody's going to want again. So there's a huge number of different steps being taken now to ensure the safety of bystanders, including clinicians. And that includes global guidance from different critical care societies and different groups that would suggest you need to maintain a closed circuit during mechanical ventilation, and that instantly affects your choice in terms of device type, okay? So PMDIs, jet nebulizers, both require opening of the circuit in order to deliver drug or to refill the drug. And of course, you're looking for patient outcomes. So you want a device that you know is safe for the patient, safe for the healthcare professional, but also is actually going to lead to proper patient outcomes. And as well as that, and what follows in this world is the healthcare economic side of it. And again, we're not talking always about drug-device combinations, but there is the opportunity for drug-device combinations in mechanical ventilation, where there's quite a number under development. And certainly, again, I think many of you will agree that post-COVID, inhalation has got sexy again, and there's a huge renewed focus on inhalation as a means of drug delivery. So, but with the healthcare economics, and Wilbur mentioned, the vaccine is an example, we use 1/5 of the dose there, but there's many other drugs that would have been administered by IV over the years. Now we're seeing either reformulated or not reformulated, and now placed into a nebulizer and using vastly reduced dose volumes. And that, in some cases, can lead to savings of tens of thousands in a single patient. So that's vitally important. But also some of the work that we've done and we've published elsewhere is that by giving the amount of drug that the patient requires means that there's a better chance that they'll be sicker for a shorter amount of time, which means they'll get out of the hospital quicker, and hospital days or ventilator stay days are reduced. And when the average cost of a hospital ICU visit is about €50,000, you're looking at a significant saving there, if you can even save 1 day of a patient's stay. So in this particular study, what we looked to do was set a baseline to understand a variety of different factors that influence aerosol drug delivery during mechanical ventilation. What's the importance of device choice, formulation choice, your ventilator parameters, etc. But we've looked here to report a fairly tight selection of the data we've generated so far. So we've looked at PMDIs. PMDIs at the start of COVID, you probably all remember, all nebulizers are banned. Safest thing to use was a PMDI, and PMDIs are often used in mechanical ventilation. So what we've done is a scintigraphy study where we've radio-labeled the contents of the PMDI and administered 10 puffs into our mechanical ventilator circuit. We did 10 puffs instead of the 4 puffs simply because we had to do 10 to get a signal. And also important to note, we follow the instructions from the manufacturer to actuate the device at the start of inhalation. There's several works have been published over the years that would show that coordination with the breath is vital, and it's sensible, of course, but certainly on a mechanical ventilator. So Phillips, John Pritchard and Lucy Ardiker would have published on some of that and the importance to shake, etc. Ariel Berlinski in the States has done the same, and showing that if you don't administer the drug at the start of inhalation, or even if you don't follow the instructions like shake in the waiting period, you will get vastly different amounts coming out of your PMDI. So to be as fair as we could be, we used the standard protocol and the correct protocol. We used the Aerogen vibrating mesh, the Aerogen Solo in this case, placed on the dry side of the humidifier, which is part of that component, that system that I showed you on top there again, for active humidification of the airways. So ventilators take room air or tank supplied or wall air supplied gas, typically oxygen air, and they can tighter and add more oxygen as needs be, but it's a dry gas. So that humidification is required either by active heated humidification, which is like a kettle boiling essentially and delivering the vapor in that case, or through what they call passive humidification, which is a sponge, a HME placed at the end of the circuit so that the patient's own exhaled moisture is caught and then re-inhaled again. So there's different ways to do it. So in our study here, we used ex vivo lung taken from large white pigs. We had 2 groups of 3. Again, this is part of a larger study, but 3 animals for vibrating mesh, 3 animals for PMDI, and they were in the weight range of 102 to 126. So I happily, not so happily, sit in the 110 range. So I'm in between there. So I have a decent idea of how much drug I might get if I'm ever ventilated, which hopefully won't be the case. But you can see here we have the lung suspended in a bath of warm saline. And that allows for proper recruitment of the airways. Oftentimes you'll see these lungs laid out on the bench and there'll always be atelectasis, there'll always be airway collapse, simply because of the weight of the lung on top. In this case, using the water bath or the saline bath here helps mitigate a lot of that. We used a mechanical ventilator in volume control mode. Tidal volume delivered each time was 800 mils. The reason we chose a single fixed volume is because it was the fairest test. We knew that the variability introduced by each of the lungs wouldn't have so much of a bearing, as opposed to if we went pressure control, each lung would deliver a different volume to reach a different peak pressure. So in this instance, it's a properly controlled study with adult mechanical ventilation settings. And obviously, we've done scintigraphy under gamma camera. We did 120 seconds for the vibrating mesh because we got a very strong signal, and 360 for the PMDI, just to get a strong enough signal to be able to do our maths. Now, one of the things that we mentioned, or I mentioned, was the patient safety element of it. Not only do you want to deliver a safe breath at a safe pressure and a safe volume, you also make sure that the dynamics of that ventilator over time don't change. And one of the ways that that can change is that the filters on the expiratory side get wetter and wetter over time. Now, it's not because of the nebulizer, it can be because of the patient's breath, and it's going to be because of the humidifier. But often, nebulizers are blamed for that. So what we did was we had a look at that whole system, including the lung resistance to essentially exhalation at the very start, the middle, and the end of a dose, just to see could we pick up on any increase in resistance. And the answer was essentially no, which is a nice comforting thing to have, or to have in the back pocket, because there's very little data on that. And even clinically, whilst it's very easy to read it off a ventilator, nobody does it. It's hypothesized that it's the nebulizer because we didn't use it last week and that patient didn't see an increase in resistance. It's very patient specific, and it depends on a lot of different factors. So in this instance, this is the scintigraphy results that we have. So on the top, we have the vibrating mesh and the results for each of the 3 animals. And down the bottom, the 3 animals we included in the PMDI group. So what you'll see here on the right, we got about 11.5% of the initial dose placed in the nebulizer delivered to these lungs, and with the vibrating mesh. And with the PMDI, we got 1.3. Now, that 1.3% seems very, very low, but it has been published and has been predicted by some of the bench models that we've seen used over the years. But the interesting learning here is not so much about the percentage, but it's about the micrograms of drug. So you're looking at same patient, essentially, same ventilator settings, not the same lung, same drug, but 2 different devices, and you're getting a significant difference in the amount. And considering that that was after 10 doses, not the prescribed 4. So really, you would have got 4 micrograms if that was the case, if you were to follow the correct procedures. But that's interesting, and the reason that's important as well is, and we often get caught up in talking about percentage of delivery, etc. Technically, in the end of the day, it's how much drug is delivered. And I'd very much rather be the patient getting 600 micrograms of a bronchodilator as opposed to 4 or 10, as the case may be, right? Well, in the critical care arena, you're critically ill. You need to get the therapy that you have been prescribed. And in this case here, we're just showing that device choice is critical. But also a really interesting thing that we've picked up on, and this hasn't been properly described at all in the literature, and certainly not for mechanically ventilated patients, is that ratio of tracheal to lung deposition, right? And so what you can see here is that for, we have a 1:4.25, so we get 1 in the trachea and 4.25 in the rest of the lung in terms of distribution of our aerosol, compared to 1:0.8. So we actually get more in the trachea with the PMDI than you do in the lung. And that's important, and the reason that's important is because you don't see them packaged in PMDIs, but certainly antibiotics, you don't want to be relying on a device that's going to deliver only to the trachea and only in tiny amounts, right? And again, this is not to tell you vibrating mesh is better than PMDI. This is more to understand what are the critical features that go into proper and considered either patient therapy, aerosol therapy strategies, or even just drug-device combination. So one of the interesting things we did there, taking the learnings of an ex vivo lung model, and the important thing to say was, if I can go back, we have also established that with this model, certainly with the vibrating mesh, that we sit nicely in between the previously published data in humans. So there has been human study done with the same nebulizer, mechanically ventilated adult human, and we're within that range. So we have a good model, and model we're quite happy to work with. And there's other ex vivo models as well that demonstrate similar data. But we all talk about in vitro, in vivo correlations, but in this case, we had the opportunity to do an in vitro ex vivo correlation. And we publish regularly on using bench models, because they're a great tool, but that's all they are, they're a tool. right? And the ability to include a filter at the end of a simulated patient setup in the lab is great. It gives you an indication of what's a good system, a bad system, but you should never be using it as absolutes of delivery, right? So what we were able to do here was, again, set up our ex vivo lung model and work towards understanding whether or not we got a filter. Did it overestimate or underestimate? Because filters can't exhale, right, where lungs can. So in this case, we definitely showed that filters do over-report the amount of drug that's being delivered over a lung, but you have to remember that that filter can't exhale, whereas the lung can. So interestingly, as a model, when you add the filter or lung data with its exhale data, the EXP there, we actually get the same amount, right? And so that's interesting for us because we know our model is solid. We know our model is good at picking up where the aerosol has gone, which is unsurprising given its scintigraphy. But it also gives us now a feel for what sort of overestimation you would see in using bench models. And that's vitally important, simply because bench models should be used as tools to give you an idea of trends, not as absolutes. So finally, the implications of this is, for us, this work at least, and we're very much focused on mechanical ventilation. And over the last 3 years, certainly, we've thrown everything into the human lung during mechanical ventilation. You can imagine with COVID, whether it be, anti-inflammatories, antivirals and everything, frankly. But this kind of learning and understanding gives us a huge amount of insight into how to best design a system for drug delivery in a mechanically ventilated patient. It also, with the data now that we have established for PMDI and mechanically ventilated system, previously, kind of early, before vibrating mesh came on the market in 2003, PMDIs were always the same as nebulizers. Bronchodilation levels in mechanically ventilated from a PMDI to a nebulizer were the same. Don't use the expensive nebulizers, use the PMDIs. Yes, but PMDIs also deliver exactly the same amount as jet nebulizers, so no wonder they were going to get a similar bronchial response post-administration of bronchodilators. So now we're going to either have to change vibrating mesh nebulizers to something else and not call ourselves nebulizers, or do a bit more work like this. But also the interesting thing here is that ratio of tracheal to lung deposition, that gives us an understanding of, okay, well, maybe PMDI is okay for bronchodilation. However, if I'm looking at anti-infectives such as antifungals, antibiotics, even just some of the monoclonal work that we've done in some of the trials, now you want that distributed throughout the airways. And again, now we've a better understanding of what facilitates greater deposition and distribution throughout the airways. Next steps to finish out the studies using jet nebulizers and complete the trio and understand a little bit more. And some of the work we've already started, should I say, is with these lungs, dissect them into the lobes and understand what's the amount of deposition you actually get in each of the lobes. So don't forget, the whole lung is the whole lung, but there's lots of things going on in there as well. So each of the lobes, and this is vitally important. We all know that those lobes won't all be ventilated exactly the same way, but as a result, they won't all get the same amount of aerosol. But there are some instances where you may wish to treat the upper lobes for whatever reason, and maybe just infection. If nothing else, this confirms that using a vibrating mesh, we were able to target all of the lobes and get some drug into that at least. And then finally, the translational element of this is very much moving towards, well, we see now some studies out there that are looking at BAL or ELF to take samples of administered Clistin, for example. And they're looking at serum blood levels and saying vibrating mesh delivers more to the serum than it does to the upper airways. But there's a reason for that. And once we get a little bit more information around the jet nebulizer data and the ratio of trachea to lung, we'll be able to comment a bit more on that. And so with that, I'll finish, and apologies for speaking so fast, but 10 minutes is not a long time. And if you have any questions, but I'd like to thank my collaborators in this, and Laurent Vicelio's at DDL here this year as well. It's always great to collaborate with Laurent, and we've done some fantastically interesting work here. And happy to answer any questions you might have.
Good. We are a little bit over time, so we have time for one quick question, because we need to catch up a little bit, unfortunately, also because people are waiting for beers, but we've got one question.
Okay, thank you, Chantal Darquin. Very nice talk. I just have one thought. When you presented those deposition in the different lobe, you have an ex vivo model,
Yeah.
meaning that the flow distribution is probably not representative of the flow distribution when you're in the rib cage with the pleural space and the intrapleural pressure difference. You're going to have more flow going to your upper lobe in an ex vivo than you would have.
Interesting. So we did consider that, and that was part of the reason why we did volume control ventilation, so that we would fix the volume. Now, 800 mils in a 110, 120 kilo animal, or human for that matter, might actually be on the low end of some of the volumes that they might be delivered. But we've through other projects, exactly that question came up, and we've done some work where we've constrained the lung and added or reduced the compliance, and interestingly, we see similar.
Okay. Thank you.
Good, many thanks.
No, it's all my pleasure.
What I would like to suggest is to all our, for all our speakers as well, is we've got a reception after this one, so if people have got questions and want to speak to all our speakers of the entire day, please contact them at the reception.
Lovely. Thank you very much.